revision about the salivary glands. The salivary glands, they are the organs that are associated with the digestive tract. So our oral cavity is the first organ that is starting the digestion. So when we say it includes the salivary glands, other glands as the pancreas, the liver, and the gallbladder. The main functions of saliva that's produced by the salivary glands are wetting or moisturizing, and secondly and secondly the lubrication of the oral mucosa and of the ingested food. So to initiate the digestion of carbohydrates, lipids by means of amylase, lipase activity enzymes, and for the carbohydrates, the amylase, and for lipids, the lipase. This is number one, wetting and lubrication. Number two, secretion of the germicidal protective substances, immunoglobulins, antibacterials, lysozymes, lactoferrin. Okay. Also, the saliva has very important role in the. Let me see here. The saliva has a very important role in the protection. Okay. Buffering function and protection of to the teeth, the enamel in the teeth, because we have the calcium binding proline rich salivary proteins. So it is very, very important. We don't have this thermo protection as in other species. The salivary glands are produced by these three major glands. The saliva, is produced by these three major glands and the minor glands that are found overall in the oral cavity. Finding that it's a complex liquid that's produced, it has digestive, we said, lubricating and protective functions. So we have, in addition to the ones that are minor in the scattered throughout the oral cavity, we have the three pairs that we will talk about them. In humans, the minor salivary glands, it's only 10%, we said, of the total volume of saliva, while the other is from the major glands. 70% of the mucus, the minor salivary glands, is mucus secretion. So we have all around these glands is a capsule. That's not present here. We have all around it as a capsule. Okay, surrounding all the glands that is rich with collagen fibers. It surrounds the large salivary glands. We have a parenchyma that consists of secretory cells, and we tell them acini or end pieces, and the branching duct system. It's arranged in lobules and separated by connective tissue. Okay, that turns out from the capsule. It originates from connective tissue that is divided from the capsule, the septa. We call them the connective tissue septa. The secretory end pieces has two types, either serous, either mucus, and we have some non-secretory parts which are called the myoepithelial cells. The secretory portion is followed by a duct system. Here you see that it's a duct system that modifies the components of the saliva and conducts it to the oral cavity. Let's start by serous. Here you go. There is the secretory. The serous, generally, you know the ultrastructure of it, but generally we have pyramidal in shape, broad base resting on the basal lamina, narrow apical surface, short irregular villi that face the lumen, and the exhibit has characteristics of polarized protein secreting cells, the serum. Adjacent cells, secretory, are joined by desmosomes. Usually, we have a spherical mass of cells here, a spherical nuclei, and a small lumen in the center. Okay, the structure can be grape-shaped, we've said, the grape-shaped appearance, all of well, and the stem is the spherical shaped, and the stem is the conducting system, the duct system. So the mucus cells, in contrast, we have cuboidal columnar in shape, pyramidal, but more like a columnar, columnar cuboidal. Okay, the nuclei here are oval and pressed toward the bases of the cell. So we have exhibition of characteristics of mucus secreting cells. They contain glycoproteins that are important to moisturize, moistening and lubricating. Okay. Most of the glycoproteins, called mucins, that's the name, 70 to 80% are carbohydrates sites, okay, in the structure. Mucus cells we have mostly organized in tubules, and they consist here a cylindrical-like lumen. The lumen here, yes, is more cylindrical, while here it's more rounded. The lumen of the mucus is larger than the serous. This is more spherical, this is more columnar, the lumen. The submandibular and sublingual glands, we have both serous and mucus. We call it the characteristic pattern as mixed. Okay, so the ends are ends of the mucus end pieces are capped by serous. We call them demilune. Okay, they appear as demilunes. The myoepithelial cells are found between the basal lamina and the basal membrane of the secretory cells, secretory end pieces, and also the intercalated ducts. Okay, to a lesser extent the ducts, but most likely the end pieces, secretory pieces, with which these form the initial portion of the duct, the intercalated ones. These cells surround each portion, 2 or 3 usually per secretory unit, and they are well developed and branched. We have processes, some call them basket cells, we said, and those ones that are associated with intercalated ducts, we have them as spindle shaped. We have them along the duct, the length of the duct. The special characteristics, we have the small muscle, smooth muscle cell appearance, smooth muscle cells, so they have contractility function, as you see here. We have, however, other functions like intercellular junctions between themselves, and we have that joins them to the secretory units by hemidesmosomes. Okay, desmosomes and hemidesmosomes. The contraction is the main function, while we have the prevention of the end pieces of distension, okay, during secretion. So we need to increase the intraluminal pressure. These cells contract to make the flow of saliva out of the secretory units. Okay. Finally, the duct system. So secretory end pieces, we have the small intercalated ducts that empty. These are cuboidal cuboidal epithelial cells, while we have they have the ability to differentiate and to regenerate, okay, into the secretory and the ductal cells. These short intercalated ducts, they join larger striated ducts which are characterized by striations, radius striations that extend at the basal part of the cell to the level of the central nuclei. We see in the electron microscope the striations here are seen that have a lot of enfoldings on the basal membrane with numerous mitochondria, we've said, okay? We have lined parallel mitochondria that increase the ion transporting cells because they are ion transporting cells and we need active transportation. The intercalated and the striated, they are both called intralobular because they are located within the lobule. Striated ducts, we said, they drain into, they modify the saliva, and they become interlobular when they open to the excretory or the collecting ducts. We call them interlobular ducts, excretory ducts. Okay, initially they are lined by pseudostratified or stratified cuboidal epithelium, and until reaching the surface, where we have the main duct, it empties to the oral cavity by non-keratinized stratified squamous epithelium. Okay, all of them, they have they open into lining epithelium. We have vessels and nerves that we said they are in the capsule, the connective tissue capsule. And we have a rich vascular and nerve plexus that surrounds all the secretory and the ductal components of each lobule, capillaries for secretion, and the autonomic nervous system, we said, parasympathetic stimulation for the food, okay, usually. And the sympathetic stimulation is produced for small amounts of, produces a small amount of mucus saliva. Okay. This is mostly it. The development was by invagination or proliferation, downgrowth of the epithelium. Now the duct system is epithelial in origin, ectoderm, while the secretory portions have their development, we said, from the endodermal, more like the pouch. Okay, so this is the exocrine gland formation. Minor glands, but I want to say more about the major glands. The parotid gland, we have a acinar gland, okay? The parotid is, serous gland. It contains rich in proteins, okay? Amylase activity especially. So it is responsible for hydrolysis of the carbohydrates, ingested carbohydrates. And we have this digestion that starts in the mouth. And it continues for a short time in the stomach before the gastric juice will acidify the food and to decrease the amylase activity. So all the way we have the amylase activity until reaching the stomach. The intercalated and the striated and the parotid are easily observed. We have long duct system of intercalated and striated ducts in the parotid, and the connective tissue capsule has plasma cells as normal and lymphocytes to protect, okay, especially the parotid gland. The plasma cells, they have IgA immunoglobulins. We have secretory components with IgA-rich that is release saliva into the resistant enzymatic digestion, okay? So against pathogens, against defense mechanism against the pathogens in the oral cavity from the serous parotid gland. We have the second is the submandibular. Submandibular is also branched tubular acinar, and we have it mixed serous cells. Mostly 90% are serous and 10% mucus, almost. Okay, mucus tubules. So we have a presence of high enfoldings that increase the ion transportation. So we have in the submandibular more of the striated ducts that facilitate the electrolyte and water transport. Serous glands have weak hemolytic activity, while we have the mucus glands that have the serous demilunes over the mucus, and that produce the amylase. Okay, the lysozyme. Sorry, lysozyme, which ingest the lipids. We have also the lactoferrin, the bacterial antibacterial activity, and we have very short intercalated ducts, more like striated ducts, and then open to the oral cavity. Now the sublingual glands, like the submandibular, we have branched tubular acinar. It is predominantly mucus, the sublingual, and we have exclusively, we have serous, small portion of serous demilunes. Okay, we have the, they secrete the lysozyme. These ducts, we have intralobular, more ducts of the intralobular are really not developed. We have more the interlobular part of the duct, okay? That is after this one, the excretory duct. Where is it? Yeah, the minor salivary glands, this has a whole summary that I registered already. The minor salivary glands, they are non-encapsulated, we've said. They are found distributed in the submucosa all through the oral cavity. The saliva here produced by small groups of secretory units, and it's conducted directly to the oral cavity by means of the main duct. Okay, we have variations. Usually they are mucus, because we have here is the duct system is short and it doesn't modify the short ducts that have little modification of the content for the mucus saliva. Okay, although we have it usually mucus, we have some serous present, especially in the here posterior part of the tongue, in the serous, the von Ebner's glands. Okay, von Ebner's glands, this is the only exception. And we have the also the production of the immunoglobulins normal that are present in the minor salivary glands also, IgA secretion. This is finished. We've said also about the, here you go, the branching. And we've said that xerostomia, or dry mouth, is a condition that makes it difficult for chewing, difficult swallowing, tasting. More dental caries, more atrophy, because we've said all the functions of saliva. So when we have a dry mouth, for example, because of systemic conditions, because of medications, radiation therapy, diseases like Sjogren syndrome, okay? Here we have Sjogren syndrome is a chronic autoimmune disease that has lymphocyte infiltration. Okay, so we attack the acinar cells, the exocrine glands, particularly the salivary glands and the lacrimal glands. These clinical features will have almost a drying of everything: the skin, the eyes, the oral. The oral cavity, all of it is dry. Okay, this is the end.